Hi everyone. We'll start with our more detailed description of the electron transport complexes. In this video, we'll start with complex 1, which again, what does it do? It takes electrons from NADH and passes them on to Q. So it reduces coenzyme Q from the ubiquinone to ubiquinol state. In doing so, of course, NADH is oxidized to NAD+, coenzyme Q becomes reduced to ubiquinol, and the all-important protons, 4 protons from the inside of the mitochondria, from the P side, they will move out. Okay, and so the complex 1 is electrogenic. It will it will generate a proton motive force. This is an extreme, so it's a large complex. As I said, it's at least 45 proteins in most species. And it's actually the complex for which we know how the protons move, okay, from one side of the membrane to the other. So we can see literally the conformational changes that are occurring when the electrons are moving around, and how the protons now, that is coupled, all these conformational changes in this giant complex, how that affects proton movement as well. So this is from the cover of a very prominent journal, Nature, and this is the structure of complex 1. This is the membrane, and this part is embedded in the membrane. Okay, so that will be the inside of the mitochondria, and on the other side of the membrane will be the outside. A schematic was known already from the same group in Cambridge in England, and this is how it works. All right, so it looks like this. So the architecture, this is bacterial, but it's actually very similar with our own complex 1. This complex are extremely highly conserved, obviously, in evolution. So we have NADH, and then you have the flavin cofactors. You have FMN here, and you have a whole series of iron sulfur clusters. Okay, so the electrons come from here, and they bounce around to all these iron sulfur clusters, all these cofactors that we discussed earlier, and Q is at the bottom of this cofactor chain, let's say, of electron sulfur clusters. And these are other components of complex 1, okay, other proteins. And what happens is, as the proteins move, as the electrons move from one, they bounce around from one sulfur cluster to the other until they find Q, ubiquinone. Then, okay, that generates conformational changes, and some protons that were poised and they knew somewhere more or less where they would be. Because of these conformational changes, notice how let's say in this pink component here, it changes from this state, open from the inside to open to the outside. Now the protons find themselves on the outside. Okay, and we need to explain 4 protons here. Okay, so then the lab published a high resolution structure, and this is ongoing. Okay, more and more higher resolution structures will come about for this and other complexes as well. But this is the best understood, and this is really how it happens. So in the oxidized state, so this is how it is, more or less what we described earlier, and they had pinpointed the specific protons and where they are, okay, or where they should be, based clever deductions in the structure of all these components of complex 1. And that's, again, what you need to see here, which is actually very cool, is that where the electrons will come in from NADH to the flavin cofactor and all these other iron sulfur clusters, they're in one side of the protein, but the protons come out from other parts of the protein. But the key is to couple the movement of electrons with a movement of protons, right? And that is coupled through the conformational changes we're discussing. So as the electrons move, essentially what happens is Q gets pushed up in this at this position in the complex, and as a result, what you really have is almost like dominoes. Okay, these blocks here are alpha helices, and then as Q goes up, then it tilts this alpha helix here, and that pushes all these other domains in the other parts of the complex. And now the protons almost exactly like again, a series of dominoes. And now the protons can actually find their way on the outside. And this is how electron transport is coupled to conformational changes and to net proton movement across the membrane. It's a beautiful uh, result. I mean, it's an amazing thing to see the general complexes and the biology being fully explained with really high order protein structure determination here, right? So, and this lab, the Sazanov lab in Cambridge, keeps producing. I don't know if it's in Cambridge actually. I think he's at the Medical Research Council, the MRC Institute. Um, high resolution structures of these respiratory complexes. Now, the complex is not easy to put together. This is the final product, okay, and that's how it looks like. As you know, this part, the whole complex, looks like this. But who puts all these little pieces together? So there is for each one of these complexes, there are big assemblies. There are assembly pathways. You put them in order, and one protein after another in a particular sequence and order until finally you generate a complex. Now it turns out diseases in people, okay, you cannot live without a complex one. So complete loss of function of these complexes, any one of these complexes is actually lethal. Um, but there are diseases in people. There are all these mitochondrial diseases that are associated with the electron transport chain, and usually, how do you see? They're devastating diseases for the most part. I mean, the children are born. I mean, the babies are born, but sooner or later, at some point, the individual will die. The patient, how soon or how late, that depends on the severity of the defect, and they all have the same phenotypes, the same disease phenotypes, the no exercise tolerance, I mean, zero tolerance to exercise. And in the end, it's horrible because you can't move your muscles to breathe, okay, in a sense. Um, so, but you can see some of these mutations, and most of them for complex one happen to be in the assembly pathway of complex one. So there are various diseases in people, in human genetics that are associated with a defective assembly of complex one, which leads to all these mitochondrial style diseases. All right, so we'll close here, and then we'll continue with the next video with complex two.